finish off on one or two people.
Yeah.
You mentioned Gabriel warmly.
Yeah.
The other people who I've interviewed might be in your sort of on the edges of your field. One was Robert Hind.
Yeah, Robert is a super person. Absolutely.
Tell me
He's
about
been very positive and very encouraging and very helpful. Again, my research overlapped a little bit with Robert's because he was interested in mother-infant relationship and family relationships. And I've been very much interested in mother-infant relationship at a mechanistic level in terms of how the brain works to regulate that behaviour, and the importance of hormones again in maternalism and maternal behaviour. And now I'm talking about animals here because what is interesting about all mammals is that you actually have two generations developing in one individual. You have the infant and the placenta, which is also fetal, that actually communicates with mother to extract resources, but also to tell the mother's brain what to do. So the fetal placenta, this is fetal genome, tells the maternal genome that it's important that she takes on more food, even though she's not at that moment in those weeks in time in need of it. She doesn't need the calories because the fetus is too small, but she will need it for the last trimester of pregnancy when she can't eat enough to feed the fetus, and also for postpartum suckling. And it also talks to the brain, the placenta, to inform or prime the brain ready for maternal care and prime the mammary gland ready for milk lactime. So there's this interaction going on in utero which ensures that good mothering follows. Now, what is interesting about human behaviour is that humans don't have to go, women don't have to go through pregnancy and parturition to be perfectly good mothers. Now, I'm not saying it doesn't have it doesn't help it in ways. I'm not saying it has no impact, but it's not necessary. Whereas in all other mammals, it's deterministic. All other mammals, except for large brain mammals like primates, for example, where mothering is very much a learnt occupation. You see that monkeys will allow their older daughters and their sisters to handle their offspring. And particularly their daughters, and if the daughters get it wrong, she'll go and cuff them around the ear and take the baby back. So there's a learning process going on, and I think that learning process is crucially important in terms of maternal care, and perhaps the most important component of maternal care in large brain mammals. And one thing about the human brain in particular is that it continues to develop postnatally for 17 years, up until late puberty. Between 17 and 21 are the final years of maturation. There's no more growth, but there's a complete pruning and remodelling of the brain, particularly those areas of the brain that are concerned with forward planning and with controlling emotional behaviour. So really, this care goes on, not just in utero, as it were, but for many, many years afterwards. Most of the brain development occurs postnatally, and when you've got a large brain, then you need to be giving the kind of care that is important for that brain to develop, in other words, in the most appropriate social environment. And the other thing that interested me about maternal care is the mechanisms, the neural and the endocrine mechanisms, in particular, the way the hormones act on the brain to prime neuropeptides, in particular oxytocin, for maternal care. And what is interesting about what is mechanistically acting within relatively small brain mammals in a deterministic way for maternal care is also the same mechanism that is acting in humans and monkeys in terms of their social interactions. So that was the next thing I got on to working. First of all, working on maternal mother-infant interactions in a relatively small brain mammal like the sheep, for example, where you get the mother bonding with her lamb, and there's that important bonding going on and the mechanism of that. And then secondly, how living socially and having a large brain, and most of it developing postnatally, what changes have had to occur in brain evolution in order to encompass that? What were the changes that allowed that to happen? And so I spent quite a lot of time doing that, and it's interesting that the basic mechanism, so if you look in sheep, for example, the mechanisms are very much, and in all small brain mammals, very much driven by olfaction again. Olfactory cues are the most important recognition cues between mother and infant. But when you get to the high primates and indeed humans, olfaction plays very much less of a role. But what is important is what I call the brain's reward mechanism. How the brain's reward mechanism clicks in. It's the same in both. It's the same basic structure in terms of the hypothalamic mechanisms and the oxytocins and the receptors and the brain's reward mechanism. But what's happened as the brain's got bigger is that reward system now responds to a lot of input from other parts of the brain. It's not just regulated by these neuroendocrine mechanisms. The brain has got larger to such an extent that it's becoming self-regulatory. And it regulates itself, although in regulating itself, it has to go through a very, very, very long learning process, social learning process, to be adequate, to be able to do behaviours. I mean, we kind of think about everything we do as being straightforward and natural, but unless you've watched a mother interacting with a young child, I mean, I didn't see it so much with my own kids, but with my grandchildren, I see how much effort mothers put into their babies to get them to stand up, to get them to feed themselves, to get them to walk. The amount of positive reinforcement and encouragement, I mean, it's just obsessional, but it works. And you find that kids that have been brought up in away from home, where these children have been neglected and they're brought up in a sort of nursery, they don't learn to walk, they don't learn to talk, they just, it's unbelievable. You never think of, we've actually learned to walk. You don't think of it as being a learning process, you think it's natural, but it isn't. You have to, everything that the human brain does pretty well depends upon learning.
You mentioned 17 to 21 as the sort of age at which the brain reaches its sort of maximum.
Right.
Is that also the time by which this socialization of learning process has reached its...
reached the age of reason, I think. I think post-puberty is the age of reason, where you're less driven by your emotions, or the emotions are there, but now, instead of the emotions controlling you, your brain has that possibility to regulate the emotions.
And this is achieved, hopefully, by 17, 18, that sort of...
It starts around about 17 or 18. It's the prefrontal cortex which is maturing, which of course is a bit of a worry in modern day kids who are now coming into puberty at junior school. So they're coming into reproductive maturity long before they're coming into emotional and rational maturity.
Because emotional rational maturity still is at, what, 17, 18?
Well, the brain, yeah, that's exactly... I mean, now they've got much more insights into that with the MRI scans, and they can see what is happening. And essentially what it is a pruning and rewiring. It's not growth in the sense of new neurons forming. It's growth, in fact, neurons are dying, and the biggest increase is in the white matter, which is innervating those neurons. But I think what interests me is that biology is very conservative in that it's taken this mother-infant interaction, which is deterministic, but that, and mother-infant bonding, but that is the same mechanism which is expanded into social bonding. It's the same neural mechanisms that are being called into play in terms of social bonding and social cohesion. And it's also the the same mechanism, incidentally, which is usurped by addiction. So any kind of addiction, be it alcohol, be it smoking, be it drugs of abuse, cocaine, heroin. And what is really sad about those drugs is they also destroy the maternalism. So heroin addicted mothers, usually within within a year of birth, something like half of them are taken into care, and by the time they reach school age, it's 90%. When I say taken into care, it's not always into state care, but by a relative, by grandmother or mother. So the next thing that I became interested in research-wise was trying to figure out how things are put together at a molecular genetic level. And of course that is an enormous task, because there are so many genes that you could pick on. How do you pick on them? How do you know which ones to go for? What is the important thing? Well, I was lucky in a way, because I knew Azim Sarani, and he had just discovered genomic imprinting. Well, he discovered it, but nobody believed him. And he discovered it in the context of the placenta. And I was interested in it so far as the brain was concerned, although there were no imprinted genes at that stage. So what we did is we got the whole of the genome imprinted. Imprinted genes are normal autosomal genes which are expressed according to parent of origin. So there are some of your genes which you've inherited from mum and dad, but only your fathers are expressed. And there are other genes which only your mothers are expressed. So some are maternally imprinted and silent, some are paternally imprinted and silent. So they're monoallelic rather than biallelic. And you can try and figure out what the whole of the imprinted genome is doing by making parthenogenetic or androgenetic animals, which you can do by, putting the nucleus from an egg into an egg, and you get a haploid organism of all of the genes that come from mum. You can do the same by taking out the female nucleus and putting two male nuclei in, but then it's lethal very, very early in development because of the placenta primarily. But what you can do is you can make chimeras, that is to say you can take cells and make them parthenogenetic or androgenetic, and providing they don't exceed 40% of the total, they will survive. And that has a very big impact on both brain growth, brain size at birth, and on whereabouts they're going to in the brain. And since then I've been following through imprinted genes, and to cut a long story short, I'm still interested in brain evolution, but I'm going back to that maternalism and mother-infant, and how important the matriline has been in this context. And I'm particularly interested now in how the brain and placenta have coadaptively evolved. And that's the things that I'm currently working on. I'm also working still on olfaction, because I'm interested in small brain mammals. Olfaction is the most important sensory system they have, and how adaptive that might be. How adaptive it might be according to the environment they're in, because you may not know this, but your olfactory and your pheromone, well, we don't have the pheromone receptors, but animals' olfactory and the vomeronasal pheromone receptors are continually turning over, the receptor neurons. These are the only neurons, or were thought to be the only neurons in the brain which regenerate. We now know there are some that go to the hippocampus as well. But anyway, the question is, if they're continually turning over, are they coming up with the same repertoire, or is their turnover being selected in such a way that they're better able to respond to the environment in which they find themselves? And that's what I'm interested in now. So both of these things are interesting not so much from a genetic point of view, but from an epigenetic point of view. In other words, genes which are regulated and expressed as a result of environmental influences and are environmentally modifiable and it's that epigenetic process that makes them environment. So evolution, as it were, is in part dependent on environmental, selective things, but it may not be just a passive selection, it may be much more active than we previously thought.
Fascinating. So many things that arise out of that. I mean, just on a lot of your description at the end reminded me of your early interest in chess. I mean, the complexity of solving these problems is like playing multi-dimensional chess against a very good opponent.
But the difference with science is the only opponent is yourself, because it's an intellectual challenge, and you're challenging yourself to try and get an answer, and you don't always come up with the right answer. I think that's, to be perfectly honest, but you are continuously challenging yourself. And so if you try and think about genes and behavior, well, first of all, genes don't code for behavior, genes code for the coding of the brain. How do you know what part of that brain development is crucially important? How do you know which of the genes are the ones to try and get a handle on this very, very, very big program that's going on? And so they have been really the challenging questions. How to, as it were, be able to narrow things down without losing track of the big picture. And I think that's what's really interested me, and what got me interested in this special aspect of mammalian gene regulation called genomic imprinting. So relatively few imprinted genes.
Have you, in trying to solve these problems, have you found any techniques which encourage you to, or help you to find answers? I mean, I'm thinking of very crude things, but some of the people I've talked to have, like Pat and Gabriel and so on, have said that walking together, conversations with their colleagues, or particular stimulants to making larger leaps or paradigm shifts.
I think certainly discussing things with colleagues. So if I go back to the early work, discussing things with Joe in particular, if I go to the more recent work, discussing things with Azim, and with my interest in the placenta, discussing things with Charlie. These, because Charlie is not interested, doesn't know about the brain, Azim doesn't know about the brain, but they have in-depth knowledge which I have had to generate in myself, and the easiest way to generate knowledge about a completely new area is to talk to somebody who's interested in it, because then you get, especially if it's a fellow scientist, because then you get the social enthusiasm, the contact, the enthusiasm, and the sowing of seeds that make you want to know more about this. It's not quite so, oh, this is intellectually important, I'll go and read about it. Because you do go and read about it, but you need some primers, you need some direction, as it were, and some discussion on which to go about it. I mean, Azim is the most modest person you could ever imagine. He would never forgive me for saying that he's been extremely stimulating and given
well, I'm
me a
hoping
lot
to
of
interview him
ideas.
shortly, so
Are you?
I've asked him, yes.
Oh yeah, he's lovely. He's a lovely person.
It's notable, well, perhaps it's just a coincidence, but the two people you mentioned there, Charlie Loken and Azim Surai, are both Fellows of King's. I mean, some people, Pat and Gabriel, for example, and others have mentioned that collegiate structure, as I've found, helps long-term conversations with people, because you don't need to force it, but you meet them.
That's right.
Socially and continue a conversation.
No, that's absolutely true, and I would agree with that. I would also say that supervising is a wonderful stimulus as well, believe it or not, because it forces you to keep broad. So it's no good my saying, I work on the hypothalamus in the brain and I know about that. You don't. You have to put that into context of everything else. And every year I used to find students would ask me questions in a way that I hadn't really hadn't really put it together that way before. You know, it's not that I didn't know all of the answers and all of the bits, but I hadn't put them in quite that format before. And they'd asked a question, and I thought, yeah, that's interesting. That's an interesting way of looking at it. So, and there was never a year went by when I didn't at some point get some kind of stimulus. I mean, it's always rewarding to teach young minds, but it's rewarding as well in terms of stimulating yourself and keeping yourself aware of things.
And coming back again to the college, are there any other people in the college who, I mean, Sydney Brenner, Pat Bateson? Sydney
is somebody I have an enormous admiration for. I mean, I love to listen to Sydney, and I love to talk to Sydney. He can be extremely rude, and he can be extremely put down. You know, he'll put you down just like that. But I'm prepared to put up with it because, in fact, when I was admitted for my fellowship, I sat next to Sydney, and I came away feeling about this big. You know. Oh my God, what am I doing going to King's with people like this there? But on other occasions, you know, I've spoken to him, and I know him well enough now to realise that Sydney's not a very good listener. But a few prime words, and you can get absolute monumental amount of knowledge and information and ideas that come out. I'm not saying they're all right, but you know, Sydney is just pure crystallised intellect, genius, I think. Now I have a lot of time for him.
Several of the people I've interviewed said that he's the cleverest man in Cambridge.
Absolutely. Without any, in my view, cleverest man I know, without any question of that. And I'm proud to have been at the same college with Sydney. Hey, you know. There are times when I could strangle him, but especially but you learn. If you come with a guest, one thing you don't do is you don't introduce him to Sydney. you let Sydney want to come over and talk. Then it's fine. But, yeah.
What about Pat Bateson? Did you come across him? Because he was the direct predecessor.
I took over from Pat, but of course then Pat came here to become Provost of King's. And I kind of went into Madingley to take Madingley in a slightly different direction to what Pat had done. So there was a time when he was here, and so we were not really interacting very much. But since he stopped being Provost, and he came to Madingley, he's been involved much more with myself and postdoc and graduate student, and we have been doing things together. And Pat is now very interested in epigenetics because he likes that way of being able to engage the environment in terms of you know, gene-environment interactions in terms of developing the brain and developing behaviour. So I think he's enthusiastic. And he's enthusiastic about the work that I do.
Two final questions. One was, not sure whether you fully finished off the earlier story you were telling about the similarities and the differences between human brains and other higher mammals.
Yeah.
I mean, you said they were very similar, but there was something special about the human brain, and you even linked it to perhaps to imagination or to religion or something like that.
Well, I mean, what I would link it to is the long developmental process that it undergoes postnatally. So the brain that is forming continuously in a social environment, and that is what shapes the brain, what makes the connections and what makes different connections in different individuals. So that I think is very important. Now, in terms of religion, if you like, or in terms of belief systems, we all of us need belief systems in the sense that we all of us understand what we mean by guilt and what we mean by shame and what we mean by blame. And if we didn't ever feel guilty or shameful, then, you know, you need an internal representation of self to be able to feel guilty. You need to know what you should be and what you're not being, and you're not matching up to the standards that you set for your internal representation of self. Now, some people, that internal representation of self, they put God in that place, or they put some religious being in that place to set that standard. I don't feel you need any external reference point. I think it's an internal reference point, but you definitely need that internal reference point. You can't, and that's what comes to you when you reach maturity, being able to value your relationships and value what you put into them, and value all of the things you do, really, in terms of what you expect of yourself. You know, it's so all of the things that I've achieved, as it were, I feel that I've had a lot of influence from other people, but I don't thank God for it, if you know what I mean. I think that it's come about partly through my own direction, but partly through, especially through, in fact, being in an environment like Cambridge. Cambridge is just such a wonderful place for anyone who wants to learn and be curious. I mean, it's just full of it. You could live 1,000 years and or compress your lifetime into expend it to 1,000 years and go off in all different disciplines and learn something. You'd never come to an end of it. It's absolutely wonderful. I think what you've got to be careful about is losing focus. So it's very easy to lose focus because there's so many things which are intellectually stimulating.
Well, we covered a great deal, but sometimes one misses some important point. I don't know whether, for instance, you're married or have a family, or...
I am married. My wife is Spanish, and I just thought we were here to talk about academic things. But no, the crucially important part of my life. I have 3 children who are now grown up, and the 2 daughters have children, so I have grandchildren. And that has been my, as it were, secure base from which I've operated. That has always been there. It has always been welcoming. It has always understood me. It has always allowed me to do the things that might not necessarily generate a lot of income and what have you. But it has been, well, I don't think I could have done anything without it. We all need a secure base from which to operate, and my home is my security. And I don't force my family to engage with what I do academically. I mean, if they're interested, then great. But I don't. It's not that I keep it separate, but it's just that it's a different story. It's a different part of my life, but an equally important part.
And presumably watching the interaction, mother, as you said, with grandchildren particularly.
I didn't notice it with my own children so much, I have to say, and yet I watch behaviour. But you're too involved, you're too emotional, you're too close. You can't stand back and look at it and see it in the same way that you do with your grandchildren. Then it becomes very clear, very obvious that gosh, all that time and effort and praise and cuddling and warmth, just to get them to stand up. It is. It's amazing. And then, when Dad comes home, look, watch this, and it goes over again, and kisses and huddles and warmth, and the child thinks, what have I done? With big smiles all round. So, yeah, I think that's great.
Is there anything else that you would like to have me to ask you or talk about?
I think so. I mean, do you talk about honours and awards and
yes,
things like that? I was made a Fellow of the Royal Society in 97.
87?
97.
97, yeah.
I'm a fellow, an honorary foreign fellow of the American Academy of Arts and Sciences in, I think it was 2 years after. A fellow of the Academy of Medical Sciences. Is it the Academy of Medical Sciences or Academy of Medical Sciences? The British, I can never remember these things. And I was awarded the Wiersma Professorship at Caltech University. And
Calcut.
Caltech.
Caltech.
California.
California, yeah.
And can't think of the others. There are a number of awards and honorary lectures and goodness knows what else, but
they'll be on your website.
But they're standard. They're all, everything's on my CV. They're standard for Cambridge anyway.
Yep, it's a high standard, right? Yeah.
I mean, it's when you're talking about Nobel prizes that you.
And two if possible. Anyway, thank you very much indeed, Barry. I think that's covered everything. Okay?
My pleasure. Thank you.